You are warmly welcome. So now we call on the DVC A&R, who is representing our Vice Chancellor, Professor Folashade Ogunshola, FAS. So DVC A&R, in the person of Professor Bola Obo, will give us the charge from the university. Prof. Ma, you're highly welcome.
Good morning, dear colleagues. Good morning, distinguished speakers, invited speakers at this conference. Good morning, all participants. On behalf of the Vice Chancellor, Professor Folashade Ogunshola, ONFAS, I heartily welcome all of you to the second scientific conference of the Faculty of Pharmacy, University of Lagos. On behalf of management, I want to say we are most delighted that this conference that you started, you are trying to sustain, and we are very pleased with that as the management of the university. We thank the Dean for sustaining this and putting this together, and for all the LOC members for making this a reality, despite the various challenges that we are facing in the university, even at a time like this. The theme of your conference is pharmaceutical research and innovation, exploiting the socio-economic benefits. And I will really be delighted if this can really be escalated and drilled down in your discourse today, looking at our situation in our environment, Nigeria in particular. We all know that the potential of the pharmaceutical sector in Nigeria is huge. It is large. And the local manufacturing of pharmaceutical products will really boost the national economy in different ways. Not only will it increase our GDP, capacity building, job employment, it will reduce poverty. There are a lot of things that would come out if we can really harness the opportunities that exist in our pharmaceutical sector. And we need to really address what the challenges are that is not making us be a player, even in the West African and African sub-region. If we say we are the largest economy, if we say we are the largest population-wise, what are the challenges we face that is not letting us at this time to be able to reap the benefits that exist in this sector? And I will say we should really look at the policies that are in place that would help us translate the findings of our research to be able to assist the local manufacturing industries, not only in their growth, but also to be able to do more. The general term now is the general well-being. We talk about One Health, the general well-being of the individual, of the population. And if we are looking at well-being, looking at One Health, there are a lot, the role of the pharmaceutical industry in this is very huge. And we would want to see how you want to do this. At the end of this conference, let there be definite communiques on how we want to do this. More collaboration, intense collaboration, not lip service collaboration, not collaboration on paper, but more intense collaboration between the pharmaceutical industries, our universities where the faculties of pharmacy exist all over the nation, and research institutes that are working in tandem with the pharmaceutical industries. We would want a great collaboration. We will want you to be able to come out with the policies that will drive an impact on the economy of this nation. And I hope that by the end of this conference, we'll be able to have achieved that which we set out to do. Once again, on behalf of the university management, I welcome all our distinguished keynote speakers from abroad and from within to this conference. You are joining us in the University of Lagos, the university of first choice, we say, and the nation's pride, the only university on this side of the divide that everybody wants to associate with. So you are most welcome. We wish you successful deliberations, and we say that you have a very enjoyable conference. Thank you very much for having me, and thank you very much to all the members, the dean and all members of the Faculty of Pharmacy, University of Lagos. Thank you. And on that note, I declare your conference open. Thank you.
For Professor Bola Obo. Thank you so much, Professor Bola Obo, DVC A&R, for that scintillating welcome charge. And very quickly, we want to welcome the Chairman, Planning Committee, Professor Gloria Ayola, to give her opening remarks. Prof. Man.
Good morning, Madam Vice Chancellor. It's represented by the Deputy Vice Chancellor, Academic and Research, Professor Bola Obo. Thank you for your welcome speech. Members of the University Management, University of Lagos, professors, colleagues, students, other members of the academic community, ladies and gentlemen. It's a great joy that I welcome you to the second scientific conference of the Faculty of Pharmacy, University of Lagos. We are very excited this is happening today, and we have had two postponements. So two, the National Strike last year, and the second postponement was as a result of the presidential and gubernatorial election, which took place. This conference is organized by the academic community at the Faculty of Pharmacy, University of Lagos. The first conference was held successfully in March 2021 and was well attended by participants from all over the globe. I want to assure you that this conference is not an exception. We have those speakers from all over the globe, as well as participants. The theme for this year's conference is Pharmaceutical Research and Innovation, fighting the socio-economic benefits. Pharmaceutical research and innovation play a pivotal role in healthcare outcomes, driving economic growth, and addressing societal challenges. This conference is relevant and value-adding, especially in this season when the nation is in critical need of an economic. With an annual global value of $1.48 trillion, the role of the pharmaceutical industry in boosting our economy cannot be underplayed or disregarded. The conference consists of 6 plenary sessions and 5 parallel sessions, consisting of oral and poster presentations over 2 days. We are deeply honored to have with us today as our keynote speaker, Dr. Umar Bindel, the immediate past Director General of National Office for Technology Acquisition and Promotion and Director of Innovation and Research at the Air Force Institute of Technology, Kaduna. For the plenary session today, we are privileged to have Dr. Bahija Rais Abraham, the founder of King's College, lecturer in pharmaceutics and academic lead at the same institution. He will be delivering the first plenary session on nanotechnology and drug. The second plenary session will be anchored by Professor Abiodun Ogudaini from the Faculty of Pharmacy of Afemi Awolowo University, Lagos, Nigeria. He will be speaking on the topic titled Drug Development and Discovery, a tool in national development. The third plenary session will be anchored by Professor Neeraj Brahman, who graciously hosted me during my sabbatical at King's College London in 2019. Professor Miraj Rahman is the antimicrobial research team lead at King's College, who will be talking on innovation in developing antimicrobials in the age of antimicrobial resistance. Tomorrow, we'll have the privilege of following speakers. Professor Martin Demeje, the Director General of Nigeria Natural Medicine Development Agency, who will be speaking on innovation in pharmaceutical technology. We also have with us tomorrow Professor Ifeoma Ejionu from the University of Nigeria, Nsukka, who will be speaking on economic exploitation of microbial genome. Lastly, tomorrow we'll have pharmacist Peter Elias, who will be speaking on contemporary health and wealth opportunities. Our gratitude goes to all our distinguished speakers. I have no doubt that they are uniquely placed to address the theme and sub-theme of this scientific conference. I also want to appreciate our abstract reviewers, assessors of presentations, sponsors, namely Astronaut Pharmacy Limited, Pharma Alliance, Biopharm Nigeria Limited, Victory Drugs Limited, Fitness Care Limited, and Bond Chemical Nigeria Limited. Thank you so much for sponsoring this program. I want to appreciate all our participants. Without whom the conference will not have been able to do. Thank you very much. I want to appreciate especially our Vice Chancellor, Professor Folashade Oguntola, FAS, who is represented by our Deputy Vice Chancellor, Academic and Research, Professor Bola Obo. I want to appreciate the management team of the university, the Dean of Faculty of Pharmacy, Professor of Lagos, Professor Aderunke Adepoju, members of the conference planning committee and subcommittee, and the academic community at the Faculty of Pharmacy for their immense support. I would like to encourage both to participate fully in all the conference at the promised basis. Please make of the network colleagues and students, change ideas and experience, and make new friends. I wish you a successful conference on behalf of the conference planning committee and the entire Faculty of Pharmacy. Thank you very much.
Thank you so much for that wonderful and detailed opening remark. Our dear Professor Gloria Ayola, the incoming Dean, Faculty of Pharmacy, who is the current chairman of the planning committee. If you don't remember everything that she said, you don't forget that she said, please try to network and make new friends. So very quickly, I want to recognize some people that just joined us, dignitaries, distinguished dignitaries. We have with us Dr. Ezekiel Akinkunmi, National Chairman, Nigerian Association of Pharmaceutical Pharmacists in Academia, NAPA, and other professors from universities in Nigeria and outside Nigeria. You are highly welcome. Now, very quickly, we want to listen to the keynote address by the keynote speaker, Dr. Umar Ubabreda. It will be taken by Dr. Ebele Omocholoba. Thereafter, we have question and answer from that keynote. Thank you. Citation, please.
Good morning, everyone. Citation for Engineer Umar Ubabreda. Engineer Umar Ubabreda, a Fulani man, was born in Yola Town, Adamawa State, Nigeria. He has his basic education in Yola Town and graduated from University of Maiduguri, Borno State, Nigeria, with a first-class honor degree in agricultural technology in 1983. He has a master's and doctorate degree from the prestigious Cranfield Institution of Technology, now Cranfield University in the United Kingdom. He professionalized as an agricultural machinery design and development engineer in 1987 and 1991, respectively. Engineer Bindia has numerous professional qualifications and awards. Nigerian Society of Engineers, Fellow of the Nigerian Institute of Agricultural Engineers, Fellow of the Solar Energy Society of Nigeria, and National Productivity Order of Merit. He was appointed as the Director of Innovation and Research at the Air Force Institute of Technology, Kaduna. Currently, he's a former special assistant to the President and National Coordinator of the National Society Investment Program in the Office of the Honorable Minister, Federal Ministry of Internal Affairs, Disaster Management and Social Development. Engineer Beda is a thorough technology penetration expert and accomplished engineer. His ingenuity and exquisite skills in ideas generation and innovation, project development and implementation easily stand him out in all his places of primary assignment. As the then Director General of NOTAV, he was the driving force behind the national policies and strategies aimed to link academia and industry. He is still the champion of the strategy to develop science and technology parks and clusters in Nigeria. Engineer Beda is the founder and chairman of the Beda Knowledge Center, located in Yola Town, Adamawa State. Beda Knowledge Center is a non-governmental organization established to support children and youth to acquire modern knowledge in both education and entrepreneurship. Engineer Beda's passion mainly lies in solving poverty challenges through.
All right then. Good morning. First of all, let me thank everyone that has participated to invite me. I hope you can hear me. And as you heard, my name is Umar Bindir. I'm an engineer by training. I've done many things. One of the things that I have enjoyed most in most of my career is actually the aspect of being a social entrepreneur, meaning I did things for people. One of the last things I did was in Yola Town. I bought this property and then I built this. I sold everything I had. And this edifice, I registered an NGO and I named it Bindir Knowledge Development Center International. It's not a school, it's just a center to engage young people in creativity, in innovation, in the introduction of information, communication, and entertainment technology. So in this facility, I have a hall. I have 120 computers, desktop, all of them connected to the internet. I have a visa there. And every day, I take 100 children in my center to experience technology, to now explore whatever is happening in the world. They'll have direct contact to the ICT facilities. Nobody shouts at them. Nobody beats them. If they break a mouse, I'll buy a new one. And then that experience is a continuous thing, and you can come as many times as you like, free of charge. I even go to the community to pick the out-of-school children in the rural shelters, bring them into the center, let them have experience of Scrabble. We open computers that are dead, we show them transistors and so on and so forth, and they are very happy. I am moving with this very well, but I bought the next piece of land and I opened up a sports arena and I allow the parents and the children to literally complete the cycle of all work and no play makes Jack a dull boy. So this play is quite enhances the effectiveness of the facility. And I think this innovation project is not only illuminating, it is socially fulfilling. And I welcome all of you to partner directly or indirectly with us to either deploy it in your areas or also to help us with ideas. But today, we are going to very briefly speak about pharmaceutical research and innovation, exploring the process of such to the socio-economic development. I modified the title a little bit, and I put in Nigeria so that it makes sense. The keywords in this title is pharmaceutical, which is a professional scientific field, research, which is science and technology, exploration, discoveries, innovation, creativities for enhancing the marketability, wealth creation, job creation, and then socioeconomic literally is how does it affect the social system, and Nigeria is our country. So I re-looked at it. So I said, if I am not a pharmacist, and I'm not even a biological scientist, I'm not a medical person, but pharmaceutical being a professional, practical profession, my talk literally applies to all professional areas. And the research covers the science and technology evolution. And again, my presentation will cover this broad area. Innovation is consistent. You use existing knowledge to be useful, literally. And then sustainable development is the socio-economic, and the relevance is that our own country. So irrespective of the sector, whether it's pharmaceutical, whether it's the full medicine or engineering, I think connecting research, innovation to sustainable development in real terms actually is doing three things. Once it does not touch these three things, you are just being too academic and you're doing your own thing, probably getting fame out of what you do, but the impact is not as good. So the first thing is that whatever you do in this research and innovation area must have direct connection to the reduction of absolute poverty in our country, such that people should have access to basic necessities of life. If this work is not actually leading to that, then the impact in our country where poverty is possibly between 40 to 60% of our people, it has no value. Second level, when you meet these basic needs through this work you're doing, the work should push the country to the next level to ensure that a lot of people or all the people in our country should have opportunities to lead a happy, safe, and fulfilling life. Because based on your work, whether it's pharmacy or medicine, people should end up having skills, competencies. They should be able to be very productive on jobs, and then they should be able to be gainful on that so that they live safely and securely. And then once they're busy utilizing these outputs, their ethics of discipline and respect will enhance their life, and then they'll play and enjoy and so on and so forth. And then at the highest level, your work must be able to push the country to emerge as a knowledge and a learning society built on a lot of core values such that institutions are world-class. You can be comparable to anywhere. If it is companies that you are producing based on your work, these should be global companies, branded, producing products that are everybody wants. And if it is know-how, it should be applicable everywhere. This is the three secrets. And the evidence for me, based on what I have seen and what I've done and where I've visited, is that the countries that have been able to achieve these three things, we call them the developed and rich countries, or the first world countries. Why? Because they do all these things. Their policies are good. Their universities, polytechnics, and technical institutions operate as an efficient system. And then they have all these institutions, the research we do, irrespective of whether it's pharmacy or medicine or engineering, connect together to become systems of innovation, creating things that are useful. And these countries that have done these three things, when you research, you find that their research and development investment is very, very high, averaging 3 to 5%. And when they have raw materials, they don't just cut their raw materials out of their country, they add value to them. Using the know-how of that industry, they create products. That way they create jobs and then wealth. And then these countries don't waste their time extending their work so thinly. They prioritize their technology acquisition focus, and they try to ensure that somebody leads this, and they call them a core mentor. In most of the countries, these are actually the presidents of the countries. And these countries that have been able to do these three things, they have very strong intellectual property culture. They know the value of knowledge. They turn knowledge invisible to assets and so on and so forth. And they deploy information communication, entertainment technology very, very heavily, and they have found a way to make sure that the academia and industry are actually connected based on a number of approaches. And this is not a joke. You see their key performance indicators. Their technology visibility is very high. Their know-how skills is fantastic. Their industrial products are everywhere, and their capabilities and services are well known, and they maintain a very dynamic creativity and innovation system in their country. In those early days, only Europeans and Americans are actually able to do this. Knock poverty off, ensure that the middle class is created based on skills, and then their countries are very knowledge-driven and innovative. But then many other countries, after a long time, said, no, we cannot continue. Each time we need fantastic medical things, we have to go to these Europeans and the Americans. Each time we have to buy their own pharmaceutical products. Each time we have to get their people to come and help us. So they checked very carefully, and they followed, and they learned, and they adapted all these things that those countries are doing. And today we're calling them emerging countries or the second world countries. Why? Because they too, their key performance indicators are gradually maturing to exactly what we see in the first. But then many other countries are yet to realize to follow, learn, and adapt. And they don't do these things in an organized manner, or they don't do them at all. And then these block of countries, we call them developing poor countries, least developed countries, underdeveloping countries, or underdeveloped countries. Literally, they're the third world countries. And they literally have no real technology visibility, very low productivity, high poverty, and so on and so forth. So how are all these relevant to our country, Nigeria? When you look at our country, we are a lot of people, and many of the people in Nigeria, over 200 million, are young people, English speaking, traditionally very entrepreneurial. So this is actually a very good point. If we are to do our own research and innovation in pharmaceutical industry, this particular country could be one of the top, or it can move the ladder from emerging to a first world country. We have resources everywhere, but our infrastructure is very, very weak. Our industries are weak. We now have insecurity and corruption everywhere. The poverty is very, very high. We have now emerging challenges of environment, erosion everywhere, flooding everywhere, wind erosion everywhere. But to a certain extent, we can say, yes, we're creeping slowly, and we have our democracy maturing. Now, when you look at, again, our country, you find that actually, in real sense, we have very few or no global products, our own indigenous products. Yes, we can have Nestle products. We can have Ricket Binkiza products. But these are not ours. It's not based on our knowledge. We don't really have global companies. Julius Berger is not our company, it's a Nigerian company, but the technology comes from, and so on and so forth. So this, in my opinion, based on this talk, is that we need to have a sober reflection and then reel out an agenda to push this particular sector or any other sector of relevance to the next level using research and innovation. But this is not new. If you look at all these, in the 70s, we did Operation Feed the Nation. We thought we could actually power using our know-how to feed Nigeria, to come up with agricultural industries based on our raw materials. We couldn't succeed. We did war against indiscipline. We did indigenization policy. We even, during the Abachas, we thought by the year 2010, we can actually become an industrialized country with all our pharmaceutical companies and engineering companies everywhere. That did not happen. General Obasanjo came as president, launched needs, the MDGs, some economic reforms, thinking that by the year 2020, we will be an industry. This today, it is 2023. Yaradua, may God be with him, came and launched the 7-point agenda, thinking is the smartest to actually do this. We can do this just when we limit our efforts on 7 points. That did not happen. Then the president, Jonathan, came, changed the 7-point agenda to transformation agenda. Then the Buhari regime, when it came in 2015, it was changed mantra. And then by the time he got his second level, it is next level. And he ended up leaving a policy called the lifting of 100 million Nigerians out of poverty in 10 years. So you see, not that our leaders don't know what to do. And everybody is involved here, the pharmacists, the doctors, the engineers, everybody is involved to do this. And they actually thought when they launched these things that will be very efficient and productive, we'll be globally competitive. And these good things, and at the end of the day, will evolve a very viable knowledge economy in our country. But we have always returned very, very weak scores. So why do we keep doing this with our own fantastic leaders, and we keep on failing with all these institutions that we have? To me, the core reason rotates around the understanding that when you want to score all these points to the extent of pushing your country to become a viable knowledge economy, it depends on the vibrance, the connectivity, and efficiency of your knowledge capital, hinged on a dynamic science, technology, and innovation ecosystem, the subject matter that we are talking about. It doesn't matter whether it is pharmacy or whatever. This is the master key, and there's no cutting corners. Anybody who tries to not do this dynamism is not going to win. So the basics of this lecture, to me, is that let us understand the theories and practice of this value chain, of the science, technology, and innovation value chain. So I deliberately chopped it into 3 so that we can be able to, I'm preaching to the converted, but it's useful sometimes to do this very, very arithmetically. If you look at the science house, if it is what a house, this is where you have the egghead, the professors that we have here, the researchers we have here. They're good in studying, generating high-level knowledge. They discover things, conceive possibilities, and so on and so forth. But they're specialists, highly trained. It takes a lot of money to give them and a lot of time. But what do you get from this pool of very, very highly skilled people? Yes, you can get research infrastructure. Oh, my lab is very, very good. I have the latest equipment, and my center is center of excellence. Yes, we're breeding PhDs. We're, world-class publications, and we're becoming professors. Our papers are high impact or cited in journals, but what is the relevance? All you get is high-level knowledge, quality knowledge, expertise, and specialists. If you just continue producing on enhancing this house, producing these people, you will still have to buy medicine from India because they're not as effective as you think. They're good in doing all this. So you need another group, and that is what takes you to the engineering and technology house, where people who have skills or ability to transform the knowledge from the science house into socio-economic solutions, all the prototype drugs, all the and so on and so comes from these people. They are the ones that know, oh, these are the active ingredients because the science says so. It will change the color, it will change your blood, it will do all this. These are the people, and they too, they have their own characters, but actually at this level where you're putting pilot plans or prototypes and so on and so forth, what you get, as far as I'm concerned, is intellectual property. You are demonstrating that the knowledge you have can actually be turned, a percentage of it can become intangible asset. And there are two, this is preaching to call industrial property and copyable property. But even if you have this property and you have not protected it, it is just like having a piece of land without a certificate of occupancy. It's your asset, but if somebody moves into your land, start producing, it will be very difficult for you to move him out if you cannot show the paper. So that will take you to the protection of this knowledge capital. That is the legal ownership of this intangible. And that is what intellectual property rights is all about. It comes as patents, trademark, industry, and so on and so forth, and so on, all the copyrights. And if you even have it protected, and you have the piece of paper, literally a patent is a piece of paper. It's a certificate of occupancy for your intangible knowledge. If you continue keeping it as an academic, say, oh, I have 10 patents, or I have 50 patents, oh, my patent is nice, and so on and so forth, it is only a liability because every year you have to pay a little bit of money to sustain that. Even the patent lasts only for 20 years. And it is a liability. For you. So you need to somehow be crafty to turn it into an asset to make sure that it works for you rather than you working for it. And that is the concept of technology transfer. This in itself is skill, is saying you have the source of the technology, it's all protected, move it to where it is needed. So if you have a patented process, a pharmaceutical process, and then it has become, there's a pilot to show that the drug works, and then you need to move it to a pharmaceutical company so that they can be able to utilize it. And again, there are a lot of techniques. You can self-actualize it if you have the skill. You can outrightly sell it and make money and say, take it, make money, but give me my 2 billion. You can license it, and so on and so forth. And that is what takes you into the house of innovation, where this dynamic deployment of knowledge, skills, and know-how to acquire new solutions. Sorry? Should I stop? How's my time? Hello? Yeah, let me just continue. So, you know, to apply this solution, real out products and services, and that is when you now start getting people who are involved in feasibilities, financing, training, IP management, and so on and so forth. And that is when you will start getting the products and the processes that can be sellable, industrial skills, and so on and so forth. And you start seeing products. So towards the conclusion, the Nigerian practicals on this theoretical analysis is now we have a policy. The current president said the Renew Hope agenda in security, economy, job creation, and so on and so forth. So our own science, technology, and innovation content in our country is very, very strong. Over 250 universities, and all this data is real. There's no country in this continent that has this kind of skeletal strength. Our science is there. A lot of people were in space. We have laboratories. All these universities have pharmaceutical laboratories, and so we're doing renewable energy, and so we're publishing everywhere. Our engineering and technology, yes, we have patents, we are showcasing our skills, so many creativities, and so on and so on. Even in the innovation house, we're there. People can remember Nicosan, which is a very, very effective sickle cell anemia management drug. We did it. Then we now have our own agglofluorescence hub, the Mysomate is there. We have so many of the engineering things everywhere in Nigeria. We're showcasing that, yes. So in essence, you can say Nigeria in the science, technology, innovation bandwidth, we're in the highest of technology in space. We're even down to the traditional. But despite all these outputs and creativities, the reality in our country is that 70% of our population are in rural areas, and rural areas in Nigeria are not nice areas. Why? Because this is how their housing is in many. It doesn't matter, from Bayelsa to Sokoto. This is how many we see it, even in the suburban areas, how they take their children to school. They get to the school, a country that can claim that it can export timber cannot produce tables and benches for its own young people. The water we do, I cannot see how we can get our own pharmaceutical industry at the highest level when majority of our population, this is the water that they drink. You know, our own, tools of trade. We claim that agriculture is the mainstay of the economy, producing food that we eat. But these are the core engineers. They're producing horse, and this, when we say their employment, these are the executive directors of this industry, if you can call them. Their products are very, very primitive. When their process is very manual. Our young people, when they leave school, even with pharmaceutical degrees, end up selling Chinese-made things on the road. They're driving KKs. Even if we establish institutions like NDE or SMEDAN to now try hard to push people into the innovation, this is where we let plantain fryers, vulcanizers, tailors, and so on. The list is long. And this is clear evidence of the fact that our STI and socioeconomic development is disconnected in our country. And therefore, finally, I will show you that the fundamental hidden challenge, which we have to understand, is that it's not that we don't have the science or we don't know the technology, or we don't know how to innovate. These three houses in Nigeria, they are running away from each other. The innovation industry doesn't trust the output of the science, and then the science does not have the intellectual property understanding to evolve into technology, and so on and so forth. And literally, our knowledge system on one side is not connected to the market because we don't understand that there is a value of death. This value of death is a whole science. And what to do to overcome this value of death is that we have to turn the arrows through conferences like this, inviting people internationally to discuss. When we brainstorm, we're now saying, hey, Nigerian science, come back. You have to evolve Nigerian technology, which now has to become Nigerian innovation. We have to continue to push these three houses until they overlap. That overlapping is measurable. And this is the sweet spot. Then we could have actually put the bridge so that our knowledge can move to our market. More specifically, in my opinion, to literally shock treat and practicalize this, we have to turn Institute, University of Lagos, pharmaceutical the faculty has to change from training centers and schooling system into R&D and experimentation center of excellence. You must be production and industrial oriented. You must be able to be producing enterprises that will come back and power you so that you do more R&D, and production. And this, you have to be recognized to become a tool for collaboration nationally and internationally. And if you're doing this, the kind of students that will come out of your faculty will be very innovative entrepreneurial leaders. They will have high values. They will have know-how, managerial skills, and organizational skills, to the extent that they can be able to recognize that opportunities in our country to unleash these socioeconomic things that they will find the value and the potential of natural environment, organized to produce, process, and so on and so forth. And then socioculturally, these students, it doesn't matter whether they're Yoruba, English, or Fulani, or Igbo, they will now, based on the reorientation of your faculty, become people with socio-cultural and interpersonal character to network and create strong connections, to share knowledge, experiences, difficulties, and opportunity, to negotiate and discuss and work, not to be fighting like we see nowadays. And then it is that is going to power the technical capital in them to investigate, discover, and develop things properly, and then to improve their productivity. And when these things are happening, you'll find that our financial capital will be better managed and less corruption and less unsafe characteristics and so on and so forth. And you will see that. And literally, this is what I call the triple helix, where the government, now the university, will be leading very well to power the knowledge system, and then that will feed industry with knowledge, and that is how you do that. And this is possible, in my opinion, but then we must make sure that we change our ways. We have to match what we say with actions. We have to lead and follow well. We have to be systematic and to know what we do in 2023, 2024, and we have to monitor, evaluate, and impact assess. The conclusions are literally similar to what I've said, but this is literally what I have for you. I think, but particularly in the industry, the academia should not operate just as academia. You have to have a culture of intense and proud promotion of quality Nigerian pharmaceutical technology. You have to organize exhibition and tech mart at the highest level. Not this kind of prototypes that we see in our exhibitions. We have to make sure that we develop strategic and viable partnership that engenders creativity. There should be an index from NUC to see how do faculties are partnering in Nigeria? How are universities partnering in Nigeria? We have to also build the critical mass of creative and innovative manpower by ensuring that anything we invite anybody to come and do in our country, China, India, or whatever, in pharmaceutical or anything, we attach them with people to learn very, very quickly and ensure that. And then we have to think out of the box to ensure that we too, we deploy all the science, technology, and innovation parks, spin-ups, supporting the facilities and so on and so forth, to make sure that our knowledge, each university, as far as I am concerned, should have a technology park. Your own pharmaceutical the faculty should have a park somewhere where you see your students turning out into MDs and churning out things. And then there is a new science technology policy, but two things in that is critical for lobbying. Not just allow labor to be confusing everyone. The academia too should lobby to achieve this. You make sure that you establish this National Research and Development Fund to power research. And then there is also a council, and that council should be chaired by the president to become the mentor of generating knowledge, moving the knowledge industry so that we can be able to do that systematically. With all this, I hope I have briefly discussed the issue of this sectoral research and innovation and how we can explore the socioeconomic benefits for Nigeria. Thank you very much.
Provoking and excellently delivered keynote. He emphasized that technology is a key factor for sustainable development, which cannot be obtained without research. And he admonished us to forget strife. As a country, we have to come together to ensure that our research outputs generate viable intellectual property. Therefore, he said that national innovation system, especially through viable partnerships, synergies, and networking of regulated institutions on both supply and demand. Such promotional activities, he said, must be regular and qualitative. a round of applause for our keynote speaker. Very quickly, we want to read the citations for our plenary speakers. For those that are just joining us from all over the world, this is still the second scientific conference of Faculty of Pharmacy of the University of Lagos. And if you don't know, the University of Lagos is the university of first choice and the nation's pride. Now I want to read the citation of, please, do we have questions? Since we don't have questions in the chat box, we have just one. You can also raise your digital hand and ask questions, and I can unmute you if you want to speak. Questions, please. There's a question. Okay. So I want to read the citation of Dr. Bahija Raimi Abraham. Dr. Bahija Raimi Abraham is a pharmacist, lecturer in pharmaceutics at founder of King's College London. Raimi Abraham is the first graduate and was the 2018 recipient of the Outstanding Woman in STEM Previous Award. Dr. Raimi at King's, Dr. Raimi Abraham held positions at the University College Medical Sciences Research Council, postdoctoral researcher position, and at the European Medicines Agency, EMA, as a seconded quality national expert. Dr. Raimi Abraham's research is involved in solving pharmaceuticals on malaria and latent asymptomatic infection, address the following: pharmaceutical materials and innovative manufacture to engineering using synthetic nanofibrous King's China Scholarship Council and biotechnology PhD project. Nano-facilitated strategies in infection detection and treatment using plant-mediated. This is a funded PhD project that she did. Another one is the advocacy and awareness and stakeholder engagement approach to understand and explore the global impacts of substandard and falsified anti-infectives as part of King's College London Fight the Fix. So welcome me to invite Dr. Raimi Abraham to speak now. Thank you.
Thank you so much, Professor, for the kind introduction. I just want to make sure you can hear me. I will take the silence as a yes.
Yes, we can hear you very clearly.
Thank you so much. Okay, let me just share my screen and then we can be on our way. Right, so I'm going to actually turn off my camera. Well, let's see what happens. I will keep it on for a while, and then if we have any difficulties, I will turn it off. So firstly, I would like to say thank you so much to the University of Lagos Faculty of Pharmacy for inviting me to speak today. It's an absolute honor. I thoroughly enjoyed the start of the the conference, as well as the keynote speakers talk, which was really impactful and actually made me think a lot about many things. So I'm going to talk today about nanotechnology and drug delivery. And really, I wanted to start off by also apologizing for not actually being in Lagos. So I'm currently in Enugu as part of the King's Global Partnership Fund that was successful, led by my colleague, Professor Paul Long. And we have a joint TET Fund PhD researcher, Theodora, who's in the photos, and we're here in Enugu conducting an ethnobotanical survey and also looking around the ESUT University Science, Enugu State University of Science and Technology. My hope was to be able to stop off in Lagos beforehand, but it's just not been possible. So it's an apology, but also a see you soon. Hopefully I'll be coming specifically to Lagos before the end of the year, and I'll be coordinating with Professor Gloria on that further. So, so today I'm talking about nanotechnology and drug delivery, but I've been a bit, dare I say, I've been a bit cheeky. So that's a very an honest confession, in that I really wanted to think about what do we mean by nano. And I've tried to group things in 3 areas, and you see at the last one, there's a little bit around micro, don't worry, I won't talk too much about micro here, but I wanted to highlight, when we think about nano in drug delivery, sometimes we focus more on the drug delivery systems, but there are things to consider even with the tablet manufacturing, thinking about excipients. And then when we think about excipients, then we also think about the behaviors in a clinical setting. So I should highlight, so I'm a pharmacist, started off pharmacy, then went on to do my PhD in pharmaceutical science. But I think for those pharmacists in the audience, once you're a pharmacist, you're always a pharmacist. So even when it comes to the research, I'm always thinking, okay, how does this connect to clinical practice? And so I want to talk a little bit about that, taking us through a journey on the role excipients can play, which are nanoscale as well, in a clinical setting. Then we have the sort of nanoscale drug delivery system, so nanofabrication, looking at polymer-free approaches, and I'm going to focus on the polymer-free approaches in this talk. And then lastly, a sort of a medley of drug release mechanisms from solid dispersion technologies. And the reason why I sort of group this together is because when we're looking at developing these solid dispersion technologies, particularly for improving the solubility of poorly water soluble drugs, we tend to not always focus on the release mechanisms, but what it could be. Is it a nano release? Is it a micro release? I just want to comment on that towards the end. And one thing to highlight is that with excipients, they can either make or break the success of the drug delivery system. There was a time where excipients was, I mean, these are the inactive components of a solid dosage form or a liquid dosage form. But I think there was a time where people sort of, they didn't really focus that much on excipients, but actually what we're seeing over time is not only the impact of excipient stability that can influence things, but also people developing immunological responses to well-known excipients such as PEG, that's commonly conjugated with various different anti-cancer drugs. And we're seeing people having immunological responses, certain kind of responses, allergic responses to particular excipients. So I think it's just something for us to remember. So I'm going to start off with this. So when we're thinking about excipients, I started off my journey with a well-known cryoprotectant called Trehalose. And with Trehalose, this was quite a few years back now, but with Trehalose, what was very interesting is that it's present as a dihydrate. And whilst it's commonly used as a cryoprotectant in mainly protein formulations and later on DNA type formulations, and if you're freeze drying or spray drying, most likely freeze drying, at the time it was really, there was a lot of questions around, okay, but what are the mechanisms and how do we actually dihydrate? So when you're formulating it, the water molecules that are incorporated in the structure would, you know, release. Sometimes you end up with a monohydrate, and there was a lot of discussion because this impacts the overall stability of this excipient and then overall stability of the formulation. So here we were looking at where, you know, how the water loss is impacted, and this was again to do with that micro structure, maybe even nanostructure, but really looking at the excipient properties on a very, very small scale. And just to go back to this, and you know, these transitions that we're seeing, thermal transitions, led to issues with batch to batch variation, which has impact in not only the pharmaceutical industry, but the food industry. So here we go on to, this is an example, and I'm not changing my talk, but I'm very inspired by the previous speaker, the keynote engineer, because one of the things he talked about was that collaboration, not only in your own sector as in university, but collaborating with others. And I found that really resonated. And this is an example of a case study where a clinician came to me as a pharmaceutical scientist with pharmaceutics expertise with a case, like a clinical case, because they weren't able to solve the challenge. And I'm going to talk to you about that. So this is referring to two drugs called rivaroxaban and apixaban, and these are direct oral anticoagulants, and they're commonly prescribed, commonly used at King's College Hospital and the Princess Royal University Hospital stroke. To say, so just a little bit of interjection, hospitals, the King's Health Partners that oversees like the hospital and clinical arm, and that's how we sort of collaborate with clinicians in that space as well. So they had these two drugs, and the summary productive characteristics for both drugs showed that the tablets are allowed to be crushed and delivered using nasogastric tubes or stomach or gastric tubes for the apixaban and rivaroxaban, respectively, as high. But there was an issue. One, they were having subtherapeutic rivaroxaban plasma levels. Two, having acceptable apixaban plasma levels, but the rivaroxaban was the first line treatment, so they were wanting to use the rivaroxaban. So they were trying to understand, you know, do we really know the extent and the process of drug administration via these nasogastric tubes or gastric tubes within the stroke ward? What's going on? So my first instinct, looking at this from a formulation science as a scientists and looking at things first, I was like, well, let's see what's going on. How are these nasogastric tubes fitted? So one, you can see this is the process there, but when these tubes are fitted, there's a pH check, and the pH check is if the pH, so this is sort of an solution coming out from the nasogastric tube when it's been fitted. If there's a pH of that liquid that they assess between 0 and 4, this is a good indication of gastric placement. Of 6 or higher, this would suggest placement in the lung or the intestine. And so for those of you in the audience who understand, your lung pH, I think, yeah, lung is over pH 6. Intestine, if you've got the small intestine, that's 6.8. You know, all the further down the track can go as high as to 7.2. And we know that this is important because you've got to think about the solubility of your drug as it relates to the pH. So already I was like, oh, this is proving interesting, because if when placing this, all drugs need to be placed at 0.4, 0 to 4 pH, well, that might not be suitable for all the drugs. So that could impact drug absorption and drug properties. Then when we're looking at other things that need to happen, you have the crushing of the tablets, the opening of the capsule, mixing it with water. And again, unlicensed use of medication, it's altered, it can influence the stability and so forth. So my first, so I'm taking you through my thought process and my journey. First thing was to look at the excipients. And as you can see there, I noted that in rivaroxaban, there's hypermellows in 2 areas, in the tablet core, as well as in the film core, and the film coat, sorry. And in apixaban, it's an anhydrous lactose. So rivaroxaban, you've got hypermellows, lactose monohydrate here. And then with apixaban, you've just got the lactose anhydrate, so that's a dry state. And then you've also now got the hypermellows in the tablet coat. But rivaroxaban, looking at it, you can see there's going to be it's likely to be a lot of effect when crushed. So some of the feedback that we received from the nurses on the ward was that the tablets can be quite hard to crush. And so what we looked at whilst sort of thinking about the excipients was also, okay, what's happening when you're crushing the tablet? Is there an impact in tablet orientation on hardness, which there was. But now, if we look at the scanning electron microscopy of the crushed rivaroxaban and apixaban without adding anything in it, you can already see that these excipients there's some difference in how they are when you're looking at this microscopic, nanoscopic level. And you can see that when crushing the tablets, you see more of a sludgy film, like a compact with the rivaroxaban, and with the apixaban, you actually still see particles. And essentially, when doing some studies, you actually realize that this could be leading. So the rivaroxaban is likely was stuck more in the nasogastric tube, which would then be contributing to the lower levels that are being seen in plasma. Another example, I'm just going to kind of speed through this one, but this is the repackaging of medicines. This is a common practice in the UK where medicines are repackaged and taken out of the manufacturing approved packaging and we put into something else. And this is a practice that's conducted in the UK to supposedly help with medicine compliance, so the ability of the patient to take the medicines. But you see this practice in general globally. And I do also know we see it in Nigeria as well, because I've witnessed myself where it's not so much putting it in these MCAs, just taking it out of the manufacturing packaging and putting it in these plastic bags. Whatever you're doing, whatever is happening there is altering the stability of the drug. And so we look to see what's going on these excipients, and we see that, well, actually there's a change in an influence in the drug delivery system, in the dissolution process as well of that. So moving swiftly on, so we've talked about excipients, so you can see an example of the excipients, what happens there and the impact this could have in clinical practice. Let's now move on to more of the nano world, and we look at nanofabrication, particularly with nanofibers. So nanofibers, and again, I'm just being very general here because I'm not sure who else is in the audience that may be working in this area. But generating nanofibers, the advantages of the nanofibers is high surface to volume ratio, flexible porosity, and ability for these structures to be formed in a variety of macroscopic sizes. There's still this challenge of mass production capabilities, although this has been largely improved, interestingly, because of the COVID pandemic, where there was more of a drive for companies to try and look at mass production for nanofabrication to develop the masks, and then we're being able to translate that more into pharmaceutical science. And so early work was looking at this pressurized gyration system, which had the potential to improve mass production capabilities, but at benchtop level. And then with that, we did a whole series of analysis and work there, and identified that this one system that was developed by collaborators in engineering at UCL actually had the potential to generate so many things. And this was my first time working with mechanical engineers, and I really understood the importance of being able to not just make something and collaborate, but then collaborate with the right people so that you can actually see the potential for things in a different field. If there's more time, I can talk about the discovery of the microbubbles and how that came about. And it's a classic example of interdisciplinary and cross-disciplinary collaborations, and how different people can see things from different perspectives. So a lot of work in nanofibers have focused on polymer-based. I went a little bit rogue and decided to look more on polymer-free, and this is with cyclodextrins with one of my colleagues here at King's, or there at King's. So cyclodextrins, these are generated by enzymatic treatment of starch. They're molecular structures that resemble this torus-like ring, which has an interior that's hydrophobic and exterior that's hydrophilic because of these hydroxyl groups. And here we have electrospinning. So this is another technique, more common technique to make nanofibers. And here you have where an electrical force at the tip of the needle, I hope you can see this here, just at the tip there. So this is your polymer solution in here. Electrical charge is put here, and this helps to charge the threads of the polymer solution, and in this case they then are drawn, so this would be one charge and are drawn to the collector of another charge, and as it's moving and these fibers are formed, you get rapid and solid solvent evaporation here. And then the resultant is these lovely nanofibers. You can have them in randomly aligned well aligned. And so we focused on the beta cyclodextrin here. There was a lot of screening that happened to determine that, which we don't have time to go into today. And essentially what I wanted to understand was what is the influence of these cyclodextrin concentration, solvent type on spinability, what are the key characteristics of the solution, and the physiochemical properties and characteristics of these resultant fibers. So we used 3 different types of cyclodextrins, this is the beta cyclodextrin, based on their solubility with water, and this is pending publication, so I'm not able to go into full detail, but essentially we had these 3 that we chose based on their solubility in water and organic solvent as well. And we looked at the influence of water and inorganic solvent on the resultant nanofibers generated. And what we found, one of the things, this is interesting because those who work in the cyclodextrin field will know, because my collaborator was was not best pleased with the concentrations we had to go to. We had to go quite high, and I'm going to show you that data shortly. We went above 100% weight per volume. Thankfully, I can't hear you all in case you're any cyclodextrin experts in the audience, but we did have to go quite high. But it's actually because we needed to get this cyclodextrin aggregation, because beta cyclodextrins are known to self-assemble, and we needed to form these nano-sized aggregates within any aqueous-based solution to facilitate nanofiber formation. And so you can see here where we have the different concentrations of the cyclodextrin. You can see that we're getting, you know, gradually with each concentration, we're getting something. I have to say this was one of the very challenging study to conduct, because the researcher would go into the lab and be like, nothing formed. I'm like, okay, let's stay positive, please. And you can see in some instances it took a lot of different solutions to try and get there. So eventually we were able to get nanofibers, but generally speaking, it was over 100% weight per volume. And you can see here when we do this sort of triage, you can see that with a lot of tries, a lot of goes, there was success, but not a great outcome overall. But we're still positive with the results. And so we then wanted to look at the mechanical properties of these fibers and compare them with a polymer-based. And overall, we found, Overall, we found that the cyclodextrin fibers compared to the PCL, which is the polymer fibers, were smoother and stiffer. So there's potential there for cell tissue engineering work. And then we received some funding from the synchrotron, diamond synchrotron in Oxford, and this was to actually being able to visualize and see what was going on along the single nanofiber to try and understand what was going on there. And we were able to look and see the different levels of hydrogen bonding along the fiber. And what we identified was that there were bonding changes along the way. So I'm just conscious of time, so I'll just summarize sort of the key findings there, which I have already, but I think the key thing here was looking at just trying to identify alternative methods for making this nanofibers, but alongside you've got to think about the practicalities. Having a solution that's over 100% weight per volume is not really ideal, and so further work needs to be undergone in this area. But we have got some publications out around nanofibers in drug delivery, which are free to download, and this other one around solid dispersion there. So now moving forward, still on my little diagram here. So we're still in nanofabrication, but now we're looking at metallic. And I've been quite interested in the influence of shape for many years now, and every so often I dabble into this area and then I step back out, but I'm more into it now. And so if we look at with your nanoparticles, endocytosis is the main route of entry of substances into a cell, eukaryotic cell, and can either be receptor mediated, so you can see there, or can be phagocytosis or macropinocytosis mediated. And that's generally what we assume with the spherical based nanoparticles. And when you then have something that's non-spherical, there's something that occurs called nanoparticle wrapping, which is where it's like the first process in cellular uptake, and it's greatly influenced by the nanoparticle size, shape, stiffness, and local cell environment. So this is just showing here the proposed method of cellular entry for rod-shaped nanoparticles. And when you have a non-spherical shape nanoparticle, you can have high wrapping where the orientation will influence the rate of this. This is further discussed in a review that we published last year. But where is this of interest? So as mentioned, I have a research interest in malaria. And one of the challenges with malaria is that there's several barriers for crossing, for a nanoparticle to cross to be effective. And so, as you can see there, it's got to cross through the plasma membrane, then get into the coating of the plasmodium. It's a real pharmaceutical challenge. And so we looked at the influence of nanoparticle shape and material, and to see what we found. And we did find that when you had a non-spherical shape, there was an influence, but again, further work needs to be conducted here. So lastly, I think, yes, we will end on looking at this solid crystalline films and 3D printing. So this is more of the drug release mechanisms. And this is just more of an overview. So this is some work around solid microcrystalline dispersions, but the principles of this is applicable to nanocrystalline dispersion systems, where we were looking at different polymers, different stabilizing agents. What can we add to the system, the pharmaceutical film, which are very simple and easy to make and a very good patient compliance, especially if it's for oral mucosal drug delivery. But again, when you've got your drug micronized or nano-sized, you need to think about how to stabilize it. So we looked at a variety of polymers, poloxamers, Sodiplus, to look at the influence there. And then here, just taking it one step further, thinking about 3D printing, which, you know, there's a lot of talk about 3D printing being a disruptor in pharmaceutical manufacturing. And so we were looking at, can you generate these amorphous solid dispersions, but for 3D printing? And here's just something from the study. We developed these solid dispersion 3D printed fused deposition modeling 3D printed systems, where you've got your layer by layer build with your drug and your polymer there. And what was interesting with these systems, they were very porous, had lots of holes and voids, which contributes to their drug release mechanism as well. And so here we had what we presume to be this nanoscale drug release mechanism of the two drugs. Sorry, I forgot to mention that there were two drugs incorporated, and where you get the classic spring and parachute effect from both of the drugs there, which is advantageous. And this is where the amorphous solid dispersions are of value. But you can look into that more should you wish to in this article there. And lastly, just going back to the metallic, I just have a couple more minutes there. So as mentioned, this is an overview of my research area. So looking at non-animal based infectious disease models and generating these using synthetic extracellular matrix, and then also this nano-facilitated strategies for plant-mediated green synthesis. And then I bring that all together with my advocacy and awareness work around infectious diseases, being the founder of King's College London Fight the Fakes chapter, the lead for the working group, the pharmacy working group for the African Diaspora Malaria Initiative, and also my podcast, Monday Science. And this is my current research team there. They're all lovely, Saad, Tamara, Lena, Amalia, Chi. And Amalia, the work I'm about to show was from Amalia's master's, and she's currently now doing a PhD in a different topic. And Tamara also contributed, who's funded by the NDDC. And so here, we're looking at using plant natural products to synthesize as a manufacturing tool to synthesize metallic nanoparticles. And this is where you're using the plant extracts to act as reducing and stabilizing agents there. And there's several things that can influence this. So the plant selection, pre-treatment, pH, so many different things. And we've decided to focus on the influence of pH. And what we identified, one, was with the nanoparticles, you saw different sizes as a result of the different pHs. So this is going from pH 4 to pH 9. Also, we found that there was a difference in the nanoparticle size. So you can see here a medley of shapes compared to spherical. And then also the overall impacts, which is what our focus was antimicrobial properties. You could actually see that there was an impact in the activity of the nanoparticles dependent on the pH of the plant extract. So yeah, I hope that's been comprehensive and there's a clear understanding. So we've talked about nano in drug delivery, nano and a little bit of micro in drug delivery as it relates to tablet manufacturing, traditional tablet manufacturing, remembering the importance of thinking about the excipients when or overall solid dosage forms, still go back dosage forms. We've talked about nanoscale drug delivery systems, thinking about nanofabrication, thinking about polymer-free, metallic nanoparticle generation and shape as well. And then overall, then just discuss briefly around drug release mechanisms on those systems there. So I want to say thank you to all these people here and other people that I couldn't fit in on the slide. But more importantly, I really, really would like to say thank you to the University of Lagos Faculty of Pharmacy. It's been an absolute honor to be part of such an amazing event, and long may it continue. And I really hope I get to see you in before the end of 2023. Thank you. Oh, you're on mute.
For that excellent, brilliant presentation. You will agree with me that she is indeed an expert in nanotechnology and drug delivery. And she called for interdisciplinary collaboration. I promise you that this conference is going to be very educative and enlightening. And I'm sure you are seeing it. Please, because we are mindful of time, we are going to take the plenary session through the second plenary and the third plenary before we go to the Q&A. We already have 3 questions for Dr. Binder, Dr. Engineer Binder. So we're going to take the questions together. Now we are going to call on Associate Professor Ariola Jordan, Dr. Jordan, to take the citation for the next plenary session, which is titled Drug Discovery and Development, a Tool in National Development. And the speaker is Professor Abiodun Ogundaini. Dr. Jordan.
I'm happy to note that we have over 90 participants in the Zoom room, and we're very happy to have you. Please listen as I read the citation of Professor Abiodun Ogundaini, Professor of Pharmaceutical Chemistry at the Obafemi Awolowo University, which he joined as a graduate assistant about 46 years ago. Wow. He has extensive experience in teaching, research, administration, and community studies. His research focus is in the area of validation of ethnomedicine and isolating the compounds responsible for their observed biological activity. In the course of his work, he has isolated many novel compounds and has also contributed a number of compounds to the Pan-African Natural Product Library. An appreciable number of publications in peer-reviewed journals have emanated from these studies, which have attracted a number of research grants both locally and internationally, and contributed to the training of manpower for the country in the last 14 years. In addition to his teaching and research duties, Professor Abiodun Ogundaini has received the following administrative positions in the Obafemi Awolowo University, including acting head of Department of Pharmaceutical Chemistry, member governing council of Obafemi Awolowo University, member governing board of Obafemi Awolowo University Hospitals Complex, Ile-Ife. He's a public analyst and member Institute of Public Analysts of Nigeria, IPAN, a fellow of the Pharmaceutical Society and of the Nigerian Academy of Pharmacy. He has served as an external examiner at both the undergraduate and postgraduate levels within and outside Nigeria, and as a visiting professor at the University of Ibadan, Ilorin, Olabisi Onabanjo University, Shagamu, and at the University of Medical Sciences, Ondo, where he is currently based.
Please join me as I welcome Professor. You're welcome.
Thank you. Thank you for that introduction. I hope everybody is hearing me. Can you hear me, please? Thank you.
Yes, Prof, we can.
Yeah, I want to congratulate the Faculty of Pharmacy, University of Lagos, for this second scientific conference. And I wish to thank the organizers for inviting me to give a plenary talk on drug discovery and development, a tool in national development. The outline of my talk is as shown. I want to thank the keynote speaker because he had done a lot of work by what issues he has raised. And some of them that is relevant as I go along, I will be referring to them. What is drug discovery and development? The process involved. Case study on how drug development can be a tool in national development, and challenges of drug development and discovery in Nigeria, ways out, workable templates, and a conclusion. Drug discovery and development is involved with identifying a new drug, optimizing it, and bringing it to the market. The discovery bit involves identification of the compounds used for treating target diseases. And invariably, can be done by so many methods, from natural sources, plants, animals, microbes. It can be by computer-aided drug design, it can be by synthesis, it can be by rational drug design, and so on. And the development include all processes that ultimately leads to the regulatory approval of the drug. We can subdivide it into about 4 sections. The basic research, which we as university and other research institutes that we undertake, in which we identify candidate drug targets and lead compounds. Like the keynote speaker said, most of the time, we publish this without looking at the intellectual property aspect of it. And I think this is a very good conference to actually look at that. You continue the basic research by your lead optimization, scaling up of your synthesis, dosage and stability studies, safety studies and toxicology, so that you can properly validate your candidate medicines. Then you move on to the clinical aspect involving phases 1, 2, and 3. Then your drug is sent to the regulatory authority as a new drug application. And if approved, you can move on to marketing, and then the post-marketing, which includes survey of effect after white. This whole process is a very multidisciplinary affair and takes about 15 to 20 years, costing about 1.5 billion. And if successful, the drug molecule will be under patent, which can be for 15 to 20 years, depending on the country where your patent is filed. It's a multidisciplinary work that involves so many people: the chemists, the pharmacologists, pharmacists, chemical engineers, clinicians, and statisticians. And this scheme actually gives a concise picture of everything. You have a research team formed, an objective set. Then you identify or isolate or synthesize your novel compounds. You test them in animals for efficacy or in vitro and safety in test tubes and animals. Then you choose drug candidates. Then you go into formulation, stability, then scale up synthesis and chronic safety in animals. Then you file an investigation new drug application with the regulatory agent. Then you move on to the clinical phases. Phase 1 in the healthy volunteers. Phase 2 in patients with the disease you want to treat in a small number. In phase 3, you use a very large number. Then you go to the new drug application, then reviewed and approved, then the drug is into the market. But in Nigeria, what are the gaps in the drug discovery and development process? There is a huge disconnection between basic research and translation that requires huge investment, which the public sector does not have the capacity for. The keynote speaker talked about the gap between the value between the knowledge and tangible assets. It is not only researchers linking with industry that is going to solve it. In fact, it needs a lot of capital injection. And as you agree with me, researchers don't even have that kind of injection. And there are not many people, entrepreneurs in Nigeria, willing to bridge that gap. How can this gap be filled? It can be filled through public-private partnerships, investment-friendly policies by government, as in advanced clients where drug discovery and development contribute to national development. And we're going to look at some workable templates that has been tried elsewhere and see whether we can follow them to have an impactful research in Nigeria. And using the COVID-19 vaccine case, I want us to study how drug discovery can actually contribute to national development. We know that the search for a vaccine started in 2020, and by the end of 2020, Pfizer and JAMA Biotech announced a successful vaccine, and it became useful for emergency use. It's the fastest vaccines ever developed. The one before it is only months, in the year 1960. Then the Pfizer vaccine was followed by Moderna with same mRNA technology, then AstraZeneca with Oxford University with a different approach. Why was this response not the same for Ebola, which devastated communities in Africa? Why have diseases such as malaria, tuberculosis, and pneumonia that kill millions of people, mostly in Africa, not receiving such attention? The simple answer is this. Pharma companies don't put a lot of investment where they cannot recoup their investment. And since the masses of our people are poor, the investment in drugs, like I said, takes about $1.5 billion. If they make things for us, how do they recoup their money? But for COVID-19, it affected everywhere, both developed and developing countries. And even the developed countries were even more affected than us. So it was a global emergency. So they pumped sufficient resources into both the research as well as the making of the vaccine. US Operation Warp Speed injected 10 billion to pharma companies to modernize their manufacturing outfit and even the the distribution ability. European Commission pledged $8 billion to COVID-19 research. UK government contributed to vaccine retaliating to AstraZeneca. Then, since the problem of Zika, Ebola, and SARS, a coalition for epidemic preparedness was established, and it has been launched since 2017. All these ones contributed to the fast response. And all these has prepared the ground for research. There have been about 10 to 15 years of previous research to develop vaccines on related SARS and MERS, coronaviruses. Then successful sequencing of SARS-CoV-2 virus within 10 days was done because there is already advances in genomic sequencing. I'll pause here a bit. We're not doing badly. The African Center for Excellence in Genomic Studies in Ede, Redeemers University, indeed sequenced the virus from Nigeria within a short period. But unfortunately, the kind of injection that I spoke about was not made. In fact, they had the capability to make test kits. But without proper funding, they couldn't, and we were importing test kits throughout that period. This is part of the problem we have in Nigeria, and I'll still come to it. Then substantial resources were committed from ASCA companies for technological infrastructure to manufacture and distribute vaccines. Again, clinical trials is very, very expensive, but enormous funding came in for companies through philanthropists and public funders to run trials. And the trials were run simultaneously. This is not normally the case. It's usually sequential. But because of so much money that was available, it was run together. We need to learn a lesson from here. No amount of money will help without a solid platform of basic science to build on. Like I said, US, developing countries, they were already researching into coronaviruses. There were advances in genomic studies. All these were enablers for the quick response during COVID-19. What are our challenges in Nigeria? We have insufficient interest of government in R&D in the pharmaceutical sector. Most companies in Nigeria do not manufacture, and the government itself is not showing too much interest. Yes, you might say they put some money during the COVID-19. Where are the funds? And what are the funds? What have they done? The lack of funds for sustained basic and applied research. Dearth of facilities to support research. In most of our universities, the facilities for research are already aging, and we need to move with time. Then the last one, lack of funds and incentive to translate research into tangible products. I've just given the example of a center in Ede. Sequenced virus early, were in a position to make test kits, but there were no takers, neither from government or from public-spirited individual. Therefore, that research couldn't be translated for the masses. If we had done so, we would be exporting our test kits to other countries. How do we get out of this? We have to think of innovative ways to translate research outputs to usable end products for the populace and for economic development. There has to be better funding and provision of research infrastructure and equipment. And I want to discuss 3 templates that I've seen worked, both from literature and practically. And maybe we can decide at the end of this to choose which one we will, so that we can have impactful research. The first one is creating a library of natural products, the Pineapple Experience. Pan-African Natural Product Library was formed by scientists from 7 African countries about 10, 15 years ago, and is domiciled in the University of Botswana. The objective is to build a collection of compounds that were isolated from natural products so that they can be a platform for screening, so that anybody who wants a database for screening can approach the library, take the compounds and screen. And the library is intended to be springboard for further research and development by technology and pharmaceutical industry. It has a collection of over 500 compounds from natural products, and they are available in about 10 milligrams and above, about 50% of it. Because of plant the diversity of chemical structures in plants, those collections represent about 30 different classes of compounds. And half of the compounds are not available commercially from assessments from major suppliers and catalogs. And there is a procedure for accessing the library, taking into consideration intellectual property issues. The library has been virtualized. That is, all the compounds in the library has been converted to 3D. In fact, some researchers in Canada approached us and got in touch with the library, and they screened all the compounds virtually for novel HIV-1 inhibitors using the VPU accessory protein. And 8 compounds from that library, they were effective, they were active. Then 2 compounds, then they now took samples from those 8. Then 2 compounds, ixoratanin A2 and boldin, showed antiviral activity in cell-based HIV-1 and hepatitis virus replication assays with minimal cytotoxicity. Ixoratanin A2 was one of the compounds we deposited in the library. It was isolated by a PhD student of mine, now Professor Idowu. Now, the lesson from here, considering how much has been done in Nigeria on natural product research, can we not establish such a library so that we can jointly protect our intellectual property? This is a question to be answered during the discussion or during the communique from this. The second template I would like us to look at is drug research and development approach under limited resources, the China example. China was a low resource country in the 70s, and they provided a model to accelerate new drug from traditional medicine. And that's how artemisinin was isolated. And this template, I believe, can be used as they intended it for low and middle income countries, LMIC, due to gaps compared to developed countries. We don't have the resources, we don't have the support. The effort was government-led, but with public-private partnership to promote development and industrialization. I want to stress that again. Effort was government-led. And when the keynote speaker was talking, he talked of government fixing priorities. I think that is one of the things we had to do, because then the Chinese government fixed its priority, and all their research institutes and universities at all levels were working on developing anti-malaria products. That was their focus. And there was synergy by all research outfits. They screened over 40,000 herbs and compounds from TCM books and collected 4 remedies. 10 herbs, including Ginghao, were selected for further research. Then, I need to stress this. Low temperature extraction was used instead of boiling to protect active ingredient. They followed the exact methods the TCM prescribed, that is the way it's used traditionally, so that they won't make a mistake with the active compound. Animal experiments were used for safety and efficacy tests. This is something that has been used for ages. And there is no, and WHO, I think in 2014, has said that if something has been used over time and tested without any adverse effect, then you can easily use animals for safety and efficacy tests. To demonstrate the synergy, research institutes were engaged in structure analysis, and they proposed that it contains lactone peroxide. The universities conducted clinical trials and provided convincing data for its efficacy, and pharmaceutical manufacturing facilities carried out investigation on raw materials, quality standard. We can see public-private partnership highly at work here. The templates, I won't go through it. It will be available, I think, to all of us so that we can see the template. And why can't we follow this template and have a focus so that we can make progress in Nigeria? Artemisinin, of course, was a lead molecule. It has since been modified into artemether and artesunate and artemether, which are now widely used. What are the lessons that are learned from artemisinin discovery? There is a workable template for discovery of drugs from traditional medicine in low middle income countries using scientific methods. I underline using scientific methods. The effort was government-led, and it involved a synergy among research organizations. There was an efficient information sharing mechanism to avoid repetition. In this country, we keep doing research, and at times this is done mostly across, and there's no information sharing on whatever form. Lastly, public-private partnership to promote development and industrialization. Reverse pharmacology approach for phytomedicine development, the Mali example. The term reverse pharmacology was coined in 1950s, and it was to develop Ayurvedic medicines in India and also championed in China. It's based on the fact that if humans have been using some of these things for a long time, and the remedy will be effective and safe. Isolation wasn't the first thing. It came out at the end, and it was for quality control. And it arose as a way to improve utilization of herbal remedies. In fact, Reverse pharmacology inverted the drug discovery and development model that I described first. It's like you are inviting a code. What does it entail? They developed an anti-malaria phytomedicine from Agemonia mexicana deduction. They did this by selecting a remedy through a retrospective treatment outcome. They interviewed about 952 villagers. Before then, they found out the terms that were used to describe malaria, both complicated and uncomplicated. Then they interviewed them to make sure they had malaria, then what concoctions they used, and they collated the samples, and they got 60 plants. And the 3 most used plants were now tested in vitro. And there was no need for toxicology as there were no reported adverse effects on usage. Then they went to the step 2. They escalated the dose in a clinical study and noted the dose response so that they can choose an effective dose. This wasn't just done, it was done scientifically too, because they monitored parasitemia, the temperature, and if there was a problem, the person was pulled out for better treatment. Then, after getting a dose to be used, they went on randomized controlled trial to compare the remedy to standard first-line treatment using the dose they found in step 2. It was after they have finished this that they now isolated compounds so that they can use them for marker in terms of standardization and quality control. We need to note this. Then A. mexicana contains 3 protopaparin alkaloids in similar amounts, berberine, protopine, and allocryptone, and they all add antimalarial activity. One thing we need to note is that they used aqueous extraction. When they used methanol, there was another alkaloid, sanguinarine, which is toxic. But because they're using aqueous deduction, that wasn't in what the traditional healers are using. So we need to take this on board. So an anti-malarial based on Agemonia mexicana was developed faster and cheaper than for conventional drugs. The time for development was 6 years, compare about 15 to 20 for the traditional one or classic one. The total cost is 400,000 euros, and the funding was for Swiss researchers that led it. Lesson to be learned: our governments have to have the will to invest money and to provide a focused approach or priority for this. In conclusion, there should be more government involvement to drive R&D using traditional medicine as starting point, improvement of research infrastructure for drug development pipeline, improvement in research financing, then building viable public-private partnership to transform our traditional medicines resources into products. Then translation. I've said it, universities cannot provide money for translation. It has to come either from government support or from private enterprises. So we need to find a way to bridge this gap. Then no amount of money will help without a solid platform of basic science to build on. Thank you. I thank the organizers once again for giving me the platform to do this.
Thank you so much, Prof. Professor Abiodun Ogundayeni, a professor of pharmaceutical chemistry. He took us through the process of drug discovery and development, a tool in national development. And he made us to understand that if we do not add collaboration, because this research and development is multidisciplinary affair. So we need to come together and work And he also told our country that of priority. Because in spite of our hard work, the impact is not yet felt. Thank you. Very detailed delivery. Now we call on the next plenary speaker in the person of Professor Miraz Rahman. And I call on Dr. Bukola Hussaini to read the citation of Professor Miraz Rahman. He is speaking on antimicrobial agent and drug resistance. Please be very attentive as it's very important.
Can you hear me okay?
Rahman, the School of Cancer and Pharmaceutical Sciences, King's College London. After working for 3 years in the industry, he went back to academia and obtained his PhD from the UCL School of Pharmacy in synthetic medicinal chemistry. His research activities are focused on the application of advanced computational chemistry, synthetic medicinal chemistry, and chemical biology techniques to the design, synthesis, and evaluation of novel drug-like chemical scaffolds as anti-cancer and anti-infective agents. He is the author of over 90 research publications, an inventor of 15 patents, and has more than 15 years of research experience in medicinal chemistry and biophysical techniques. Professor Rahman has been working on pyrrolobenzodiazepine-based DNA binding agents for the past 16 years and developed PDB-based cancer and antibacterial and licensed University spin-out company. He is working with Public Health England to develop chemical tools to study antimicrobial resistance and discover new ways to target multiple drug resistant pathogens. He has used a combination of homology modeling and advanced computational calculations to study bacteria and fungal efflux pumps, and has invented new classes of efflux resistant antibiotics. Photogenic Limited 2013, Femtogenic Limited 2015.
Thank you very much for your kind introduction. I was noticing some internet connectivity issues. I hope you can all hear me okay. Can you hear me okay, or is it's not very clear?
Yes, we can hear you, Prof. Loud and clear.
Okay, thank you. So at first, I'd like to thank the organizers for inviting me to give the talk. And then particularly, I would like to thank Professor Gloria Ayula, who I had the privilege of hosting during her sabbatical, who worked with me for a year at King's. So I will be talking about the innovations in developing antimicrobials in the age of antimicrobial resistance, because we are probably all aware of the challenges that antimicrobial resistance pose, and the problem in identifying new antibiotic, while a number of existing antibiotics are being continuously, we are seeing them being ineffective. So what is the landscape in that area, and what at King's we are doing to identify new antibiotics. So the outline, this gives it a broad outline that I'll be talking about the current challenges of efflux resistant breaker technology, and then I will cover the validation of the technology and how we are using the technology to develop different type of antimicrobial agents. Now, this is quite an important slide because this sets the scene very well. Now, developing antimicrobials is not commercially profitable. So this was the data that came out, what we are seeing in 2022. Because you need investment at the early stage, spin out companies, biotech, to develop compounds that can be then taken by large pharma in clinical development plan. Now, what we have been seeing, that despite not finding new antibiotic, despite warning that even scratched knees, sore throat is going to kill people because of lack of antibiotics, because the antibiotics actually underpin what is the modern medicine. If you think about anyone going to hospital for even minor surgery to childbirth to major surgery, you need antibiotic to protect from infection. Now, this data shows that in 2011, there was only about 0.2 billion pound investment from the investors, venture capitalists, in antibacterial projects. And while in the cancer project, it was about 6.9 billion, and it significantly increased over the years for the cancer projects. However, the antibacterial investment remained at 0.2 billion, which suggests, despite a lot of noise, media awareness, actually there has not been much progress done in funding new antibiotics development, bringing in new projects. So you can see that there are about 34-fold more venture capitalist investment in cancer compared to antimicrobials, because if you think in a plain situation, infection is a acute indication. You take an antibiotic for 5 days, 7 days, you get completely cleared of the infection, and you can live a very happy life without any footprint for 40 years or even more, depending on at what stage of your life you got the infection. And you will never be writing a check to the pharmaceutical company saying that, okay, thank you very much, I'm now living and enjoying my life, here is your $100 or 100 Nigerian currency. On the other hand, if someone is suffering from cancer, and if a company comes in and say, okay, there is a medicine, if you take that, your life experience will be miserable. However, it will keep you alive for another 20 years. You have to pay $100 per week to access that medication. Everyone will be selling whatever they have to access that medication. And that's why cancer pays. There is a huge profit the pharma companies make in chronic disease areas like cancer, cardiovascular disease. Now, we are seeing here that the antimicrobial resistance is going to kill about 10 million people by 2050, and we are seeing drug-resistant infection everywhere. It's no longer a third world problem. But the antibiotics, if developed, can only probably make profit in year 23, and the patent shelf life is about 20 years. So this definitely after the patent period. So what we are seeing that the pharma companies are all leaving the antimicrobial space. We have not been successful in getting any new antibiotic for over 60 years. The broad spectrum antibiotic, last broad spectrum was fluoroquinolone in 1964. So which means now it is more important for academia and small biotech to innovate and then try to bear the burden of the initial development and de-risk the project to a level where the pharma companies might be interested, but still it is not very easy. So when we started to look into how we can contribute in this landscape, how we can bring in innovations, one of the major reasons why a number of antibiotics are becoming ineffective and bacteria are developing resistant is because of efflux. So the simple mechanism, bacteria has got a number of nanomachines. They can efflux out the antibiotic out of the cells. which lower the intracellular concentration, and then bacteria can develop mutations. So we have seen that we never see from wild type to a multi-drug resistant phenotype. We always look at some efflux mutant in the middle. So if we can break these, that means it is unlikely we will see multiple drug resistant phenotype. However, efflux has been a deathbed for antibiotic discovery. There are 40 years, substantial body of research, no efflux pump inhibitor has ever reached clinic because of toxicity. The last company, the antibiotic company that shut down their efflux pump program in the US in 2016. So we started to look into these, saying, okay, can we do this something differently? So we use computational modeling a lot. We have got few supercomputers in the group, and we started to develop a homology model of all efflux pumps, because these efflux pumps are very complex nanomachines, sometimes 9, 12 different protein assembles together to make a functional pump, and it is not possible to crystallize them all. We have got very few crystal structures of efflux pumps. What we noticed that the way the pump behaves towards antibiotics or substrates and inhibitors are different. So we thought, can we use that knowledge to develop antibiotics or antimicrobial agents that almost behave like inhibitors, but they retain their own target activity? So that led to these, what we call these efflux resistance breaker technology. What you see here is the current antibiotics. We look into the efflux pump, how they interact. We know the target. So, and we are using a single molecule. So they are not efflux pump inhibitor, they are antibiotics. However, they have got these efflux pump inhibitor properties, which means bacteria find it more difficult to efflux out. So when we started this project, we say to the scientific community, and I'm again saying that the way the scientific community have approached the efflux pump inhibitor concept is fundamentally wrong. And we will never get efflux pump inhibitors that will be successful in the clinic. The reason it is wrong is we kind of got sites, we kind of had a view from, say, the success of beta-lactam inhibitors and beta-lactamase antibiotics like amoxicillin and clavulanic acid, that concept that you use an inhibitor to inhibit the enzyme and then the antibiotic can do their job. But the advantage there was that inhibitor was covalently inhibiting the enzyme so that antibiotic couldn't be broken down and they were able to work. This is not the scenario for efflux pump inhibitor antibiotic combination. So what happens if you give them together? They will get in, but the antibiotic remains an unmodified substrate, which means the pump can pick up either the inhibitor or it can pick up the antibiotic. And if it picks up the antibiotic will be simply leaving the cells despite the presence of this efflux pump inhibitor. The only way to prevent that is to flood the cell with lots of inhibitors, and so that the mathematical probability that the pump will always pick up the efflux pump inhibitor and spare the antibiotic. Now, if you do that, you will start to see the toxicity, and that has been the major problem of efflux pump inhibitor antibiotic combination. And that's the reason why it has been unsuccessful to date. So what we propose that, okay, we will not go for two different agents because there is also challenges of co-development of two different agents. We will introduce that inhibitor properties into the antibiotic so that if they get picked up by the pump, they can block the pump while they maintain their own target activity. That means they're still able to inhibit the bacterial target and kill the bacterial target. So this is what we call the efflux resistance breaker technology antibiotic or ERB antibiotic. So they are single agent, they will go in and it's fine if the bacteria picks them up because it cannot be fluxed out easily. So that blocks the pump, and that means more ERB antibiotic can block the target. So that's how we develop this ERB technology. We have used this across a range of chemical classes, and there are advantages of these because this produces new chemical entity which can be patented, a very clear IP position. It can also access some of the regulatory priority like FDA Priority Gain Act. So once we started with fluoroquinolone as the first class of antibiotic, we modeled this ERB technology on fluoroquinolone. It's one of the, there are two reasons for that. One is It was the last broad spectrum antibiotic, and we know that fluoroquinolones has a very known liability for efflux, but we have now applied this to other classes of antibiotics, and in every cases we have been successful. We have filed 2 patents, which one of them have been already granted, and we are now in the process of filing another 2 patents to protect this technology. And the work has been funded by UK Health Security Agency, BBSRC, which is one of the main research council in the UK, and Medical Research Council, which is another major research council in the UK. So now in the next few slides, what I will do is I will show you how we have validated the technology using fluoroquinolones. So fluoroquinolones like ciprofloxacin, levofloxacin, they were once the antibiotic of choice for treating a number of gram-positive and gram-negative infections, and they used to sell about 4 to 5 billion pounds a year. Now they only sell few hundred million pound a year. Cipro is a lot less. Levo annual sale is about 200 million. The reason is they're becoming ineffective, and one of the main reasons for ineffectiveness is the efflux liability and associated mutations in the target. So we worked with a panel of bacteria which has known resistance mechanisms, and both Cipro and Levo do not work against them. But once we modify them using our ERB technology, you can see that these compounds show excellent activity, and it doesn't matter whether the bacteria is sensitive or resistant, the activity remains the similar. We have then tested against about 100 clinical isolates, and in every case, you can see the MIC90 is about 0.03 microgram per ml, showing that this modification can resensitize the bacteria, and bacteria is not able to efflux them out. And even when there is a presence of mutations on the target, because of their high intracellular concentration, they're able to kill those bacteria. We have then evaluated them against a panel of gram-negative bacteria, because probably know that the WHO has got this panel of escapee pathogens, and most of them are gram-negative. We see excellent activity against Acinetobacter, Klebsiella, E. coli, gonorrhea. There are some activities against Pseudomonas is where we would like to further optimize, because Pseudomonas is one of the pathogens where it's very difficult for compounds to get in. So which we have now realized that the modifications we are making is making the compound slightly more hydrophobic, and which is having an additional issue of influx. Now, here is some slide just to show that how we have validated our technology, because we were targeting specific residues within the efflux pumps by modifying them, because once we are able to access those residues, this makes these antibiotics less susceptible to efflux. So here the model shows that we were targeting some hydrophobic residues, particularly phenylalanines, and they are able to reach that. We generated isogenic bacterial strains with upregulation of efflux pump and the normal wild type, and we see that our compounds, when there is a upregulation of efflux pump, there is no change of activity, while you can see for other fluoroquinolones which are unmodified, there's a significant change in activity. This is an assay that shows whether the compounds get effluxed or not. This is known as a reserpine growth assay. So the principle is, once you add reserpine, which is a non-selective inhibitor, you see a shift in the growth curve to the right. So if the compounds get effluxed, you will see a shift in the growth curve. We see for our compounds, there is absolutely no shift in the growth curve. The reason is the compounds are not getting effluxed. So adding inhibitors is not changing anything. And we have also seen that these compounds can block efflux of other compounds, other antibiotics which share the same efflux pump. So this can be potentially used as part of combination. Now, we are not still using combination for a standard gram-negative infection. We use combination for Mycobacterium tuberculosis in TB treatment, but the way the field is moving, we believe in next decade, in 10 years time, in most gram-negative infection, we'll be using combination of antibiotics. So these looks quite good candidate as part of the combination. So once we looked at the standard public health pathogens, we work with the Ministry of Defense here. They were quite interested in looking into Burkholderia pseudomallei, which is the cause of melioidosis, which is endemic in Thailand in some part of the world, but is also considered as a bioterror pathogen. And what we have seen that the compounds that we are developing, they work very well. particularly in intracellular environment, because this is an intracellular pathogen. So you can see at a low pH, while levofloxacin remains almost ineffective, there is not real kill, but our antibiotics show a sharp kill of these pathogens within the intracellular environment at low pH. So there is more than one way we will be able to potentially develop these compounds. As part of the further validation, because we are still considering them as fluoroquinolones, that means they have to inhibit DNA gyrase, and we see that modified antibiotics, they inhibit DNA gyrase, and they are actually better at inhibiting the mutant compared to, say, levofloxacin. So you can see clear difference in IC50. And we are also seeing that these compounds, they are not, the bacteria cannot develop the mutations very rapidly. So they have got low mutation frequency, because if you have got low mutation frequency, then the likelihood of developing resistance is low. So at that point, we got funding from King's Commercialization Institute to take it forward. So we did the developability assessment, which included looking at all the liabilities of target toxicities, and then also looking at their toxicity in animal models. And everything came up very clean. We didn't see any noticeable off-target tox. The compounds were highly tolerable, up to 400 milligram per kilogram, 3 times a day. There was no toxicity in animal. And we also have seen they have got very good oral and IV pharmacokinetic profile. That means that antibiotic can be given orally. This is the PK profile. As you can see, this is compared to levofloxacin. They have got superior pharmacokinetic profile. They remain in the blood for slightly longer period of time. Very good oral bioavailability. Excuse me. And what we have seen that these compounds also are highly effective in thigh infection model, reducing bacterial burden. So we are now at the moment marketing these assets. We are trying to either set up a spin-out company. I'm experienced in setting up spin-out companies. I have set up 3 spin-out companies, mainly in the oncology area. However, it has been real challenge because there is not real interest in funding antimicrobials. I meet with venture capitalists who say, Miraz, your science is great. If you could tell us how we'll be making money, then this project can move forward. So in the last few slides, I will be just talking about the efflux-resistant antifungal agents, because we have used the same principle in developing and modifying antifungal agents. And there is a specific reason for that, because in 2021, 2020, around that sort of time, the NIH and NIAID got in touch with us about Candida auris. So Candida auris is a pathogen which has got a 65% mortality rate in hospital environment. So it varies from 35 to 60%, but it can be up to 65%. So imagine COVID, which had a mortality rate of less than 1%, has stopped the world. And if we have got a pathogen which has got this high level of mortality and we have no effective treatment at the moment, what would happen if it causes a major spread globally? Now, the New York Times article actually showed that this Candida auris is actually spreading globally, and we are increasingly seeing cases of multi-drug resistant Candida auris in the clinic, particularly in immunocompromised patients who might be in the hospital for chemotherapy or other reasons. And one of the reasons this Candida auris is so devastating impact, because it has got a battery of efflux pumps, and that results in efflux of antifungal agents, and that's why it's very difficult to treat. So we thought this is an ideal project for us, because what we could look at how these compounds work through different efflux pumps, how, say, current antifungal agents like fluconazole interact with efflux pumps, how we can modify them to make them efflux resistant. So we took on this project and developed about 108 compounds. And here is a snapshot of some of the activities. So you can see, while fluconazole and voriconazole are not really that active against standard Candida or panel. So this is not only auris, these also include glabrata, crusade, tropicalis, and albicans. Our compounds, which are YC coded, are significantly more active, in some cases 10,000-fold more active. And this is the key data, as you can see, that Candida auris, fluconazole and voriconazole are completely inactive against them. they have got their mutations in the target, which is ERG11, and the presence of the flux pumps, while our compounds that we have developed, these are the ERB antifungal agents, are significantly superior compared to them. So then what NIAID did is supported us with the preclinical data. So they funded the preclinical work of these compounds, which included MIC testing against a larger panel of fungi, in vitro DMPK assessment, and also making it ready for IND enabling studies like in vivo MTD, oral and IV pharmacokinetic profile, multi-dose PK profile, and then in the infection model. So again, I know that this is a very busy slide. I'm just highlighting that these compounds not only work against Candida auris, they are actually work against a very large panel of both filamentous and non-filamentous multi-drug resistant fungi. So we are taking this forward. We have now received some of the key data in the in vitro DMPK and MTD. These compounds are extremely safe. They have got no known liability. The only issue that we are currently considering is that some of the protein binding data is slightly higher, and we know that sometimes for antifungal agents, high protein binding leads to lower efficacy in animal model. So we are working to reduce the protein binding, but still going ahead with the animal studies. So I'll show you here some example that we do a triage before using a full animal studies, because we believe in reducing animal use in scientific experiments, and have signed up to this NC3R initiative, which is one of the initiative of the UK Research Councils. We use something called a wax moth, which is called Galleria melanella, and we can use this for a number of infection model. And here, when they're infected with Candida auris, you can see that fluconazole doesn't really protect them. So there is no protection compared to control. But the compounds that we are developing show significant protection when we are treating them with these YC compounds. And this is a Candida albicans septicemia model, and this is a azole resistant model. And we can see when treated with one of our lead compounds, YC108, we are seeing significant protection and statistically significant reduction in mortality. So at the moment, we are again marketing these. We are trying to raise about £1.5 million funding so that we can take these compounds to clinic, complete the what we call the IND enabling studies. These studies do a high-level tox, chronic toxicity, and toxicity in 2 different animal models, as well as looking at the metabolite profiling. So we are experienced with the studies because I have done this for other cancer projects. We are still have not been successful in securing this funding, but we are optimistic, and NIH and NIAID is helping us in that process. So finally, I would like to thank my group members. The data that you have seen are all generated by my group members over the years. Also, we do work very closely with UK Health Security Agency, Professor Mark Sutton and Dr. Charlotte Hine, their team. So we don't generate any microbiology data. All microbiology data is generated by UK Health Security Agency. And I must be thankful to all the funders who have funded research over the years. You might see one familiar face in these past group members, and that's Professor Gloria Ayula, who has stayed with us as part of our sabbatical. And thank you very much for listening. And I would be happy to take any questions you might have.
Thank you so much, Professor Miraz Rahman, for that intelligent briefing. I'm sure our conferees and participants are still enjoying this conference, because like I told you, it promises to be very enlightening and educative. So we are being challenged on the next course for our research. The way research is now moving from where we are and where we are going to. Please give him your applause, a round of applause, applause to Professor Miraz Rahman for this excellent briefing. Please, questions The questions are coming in and we are taking them together. However, please give us few minutes to appreciate our wonderful sponsors. And as the products are going on, our participants for your patience. And next phase that we are entering. So when we're preparing for the parallel section, we are going to call on the plenary speakers to turn on their videos. If you are still on this call, please kindly turn on your video so please, are you still on this call? Dr. Engineer Umar Binde, Professor Raimi Ibrahim, Professor Biodo Gundaini, and Professor Miraz Rahman. Okay. Okay, she said she's able to turn on her video. Dr. Umar Buba Binde and Dr. Raimi Ibrahim says she cannot turn on her video. Please kindly excuse her, but she's still on this call. So the questions. The first one is, you need to briefly address the role of entrepreneurs to bridge the valley as the universities or research institutes cannot do this. This question is directed to Dr. Umar Bubabi. Okay, if he's not here, let's take the other question. It says, good morning, sir. It's a known fact that any country that wants to develop and stand shoulder to shoulder with big countries in the world like USA, China, and others must invest heavily in research and development. For this, we see the governments in the countries in these countries invest heavily in the area. But in our country, Nigeria, this is me, begin to see the need to in science. Okay, this is for Dr. Umar, and he's not around. So let's go to Dr. Raimi. The last question. Okay, so we're going to the last questions. As we read the question for Dr. Please hold on. Abraham.
Hello. Oh, can you? Okay, I unmute myself. Yeah. Question, please. Can you read the question? Hello. Yeah. Can you hear me, please? Yeah. Okay, so for Dr. Rahim Abraham, we have we have two questions actually. So for her, the question goes thus: from your vast experience, what works best for drug delivery in targeted therapy, silica or liposomes? I repeat, from your vast experience, what works best for drug delivery in targeted therapy, silica or liposomes? And the second part of the question states that, how do you identify all the chemical components of the extracts used in nanoparticles? The second part of the question states that, how do you identify all the chemical components of the extracts used in nanoparticles? Dr. Rahim Abraham, you have the floor, ma'am.
Thank you for the questions, and thank you for the host for being able to sort out the technical problems. So the first question around what's best between silica and liposomes. Well, those two topics aren't necessarily in my area of expertise. I have sat in on a project around silica, but I think it would ultimately depend on the overall formulation aim. So taking into account the drug of use, the stability, then thinking about the patient at the end. What's the ultimate route of administration for the patient? What's your target and focus? So, it's one of those things where, I focus a lot in solid dosage forms. So that's my area of interest, and, solid dispersions. I'm not necessarily saying they're the best, but I'm saying that, a lot of people who work in different areas might feel that their drug delivery approach is the best, but it's more to do with your strategy. Each approach can be the best for whichever strategy, but you need to be very clear on your focus and the patient need at the end. The second question around how to identify the components, the compounds, I think it was, from the extract. And I would say with this is something that we've been working on. So I was actually messaging somebody in the DMs where I was saying that ultimately it depends on your area of focus, but we use NMR as well to identify some of the structures. In our case, what we're interested in is when you have your plant extract, we want to show proof that when we use the plant extract approach and use that to make these metallic nanoparticles, namely silver, and we've got some gold silver core, we want to show that there's evidence that the plant extract has contributed to the structure, the final structure of the silver or whatever metallic nanoparticle is. So we use a mixture of things. We use NMR mainly. FTIR, though the sensitivity isn't great. And then we look at other things around, so look at the element composition of the extract. And you might find that some of the extracts contain, let's say, one element, I don't know, let's say chlorine or some other element that isn't present in other parts of the mixture. So we also use some EDR, some imaging techniques. And when you have your imaging, so SEM or TEM, and then you can identify specifically the element present on certain parts of the nanoparticle. So yeah, thank you.
And the question, I hope it has been answered, right? Are you satisfied with your answer? Okay, please, Dr. Umar Buba Binde is back. So what to take his questions now?
Okay.
And anybody that wants to also add to the answers from the panel can raise up his or her hand to contribute. That's why we spotlight all of you at the same time. In the interim, I want to read out the question of Dr. Buba Binder. Professor Abiodun Ogundaini asked, okay, that's a comment, that you need to briefly address the role of entrepreneurs to bridge the valley as the University of Research Institute cannot do. Then we have another person, Henry Emediebu. I'm sorry if I did not add your title correctly. This is what I have on your name. And he said, good morning, sir. It's a known fact that any country that wants to develop and stand shoulder to shoulder with the big countries in the world like USA, China, and others must invest heavily in research and development. For this, we see that governments in these countries invest heavily in this area but in our country, Nigeria, this is not the case. So he's asking, how do we make our government begin to see the need to invest more through budgetary allocation in science, research, and development? And the last question, please, Prof, is from Professor Ifeoma Ezeonu. And she said, I just want to thank Prof for the wonderful presentation. For us researchers, the challenge is still finding the connection to transform our research into innovation and product. You have told us that we need to do that, but which institutions in Nigeria have that role? Dr. Umar, you have the floor.
Thank you very much. On Professor Abiodun Ogundami's question, where he's submitted that we need to briefly address the role of entrepreneurs to bridge the valley of death for knowledge to people, as the universities or research institutes cannot do this. Traditionally, in countries that are very, very well organized, you'll find that universities have what they call as innovation hubs or science, technology, and innovation parks, or things like that. And you see that students from such institutions migrate to become entrepreneurs. The lecturers and the researchers participate in the provision of the know-how. The institution has a very good intellectual property management system within the institution. And gradually you will see that the institutions are actually breeding that bridge on site. In our country, when I was the Director General of the National Office of Technology Acquisition and Promotion, we triggered the implementation of establishing what we call as IPTTOs, intellectual property and technology transfer offices in universities, polytechnics, research institutes, and even colleges of education. And we've implemented hundreds of them. However, because the culture of intellectual property and the effects of corruption and secrecy and lack of understanding of IP has shortened the life of the impact of these institutions. And therefore, Nigeria must understand that if we don't domesticate the know-how on generating IPR, managing it, licensing techniques, I don't think that bridge will work out very, very well. And therefore, I encourage universities to come together. If ASU can actually put pressure on government to shake the government, close down institutions, to force them to do things, I think researchers should also find some kind of platform to say that we have to be supported to generate these kind of things as a culture in our country. Professor Henry said his comment is on investing heavily in research and development. This is, there's no cutting corners to this. Any country that does not clearly invest in R&D for development will not develop. That is just the plain thing. Our own system, the investment is very scattered. PTDF have a bit of small change for R&D. Then you have the Educational Trust Fund. It also has some money for R&D. And then each institution also budget for R&D. This is highly fragmented. But again, I think the researchers should come together. The new policy, the STI policy, has a clear conclusion that Nigeria has to invest at least 1% of its GDP in total. But I also share a different view. Every multinational company that is operating in Nigeria pays for their intellectual property through Nigeria using Nigerian foreign currency. I know that because I'm Director General of NOTAP, and billions of dollars leave this country for that. There has to be a strategy to say that each of these companies that are taking advantage of Nigerian economy must also learn to share this skill. So we have taken some researchers to visit Malaysia for their oil palm research from NIFO. I took a team to there to actually see how it's done. And they've seen it that every single nut seed of oil palm is identified by barcode. Very, very weird. So that investment is heavy, and our country has to realize that. And I think a very good submission to Mr. President to ginger this using the professional organizations might work. A lot of efforts were made. None has actually popped out any reasonable thing. Finally, Professor Ifoma said the connection to transform our research into innovation and products. You know, the universities, you have told us that we need to do that, but which institution? Well, the tools are many. First, the researchers are there. The number of papers that you see coming out from R&D institutions and universities is an index that people are working. But the quantum of all those papers, for example, the proceedings of this conference, if you are to ask a question, how much of this knowledge is actually prepared for industry? Meaning, how much IPR has been generated out of all this talk? The amount is very, very tiny. That has to be addressed. And the IP institutions in Nigeria are mainly 3 or 4. One, we have the trademark and patent registries in the Ministry of Investment. These are all lawyers, and we try to reform it to make sure that scientists are also put into that to generate that kind of thing. There are a bit of difficulty there. Then we have the Copyright Commission, and this again is led by lawyers. It is in the Ministry of Justice. So you see, 2 different ministries are carrying the 2 big, industrial property and copyright. And then you have NOTAP, which is located in Ministry of Science, Technology, and Innovation, which is running the technology transfer. We tried as much as possible to coagulate all these institutions into what we call as NIPO, Nigerian Intellectual Property Organization, to mimic WIPO. But the fight on who leads it, which ministry will host it, did not allow it to fizzle. So scientists will have to resolve and also pursue these approaches so that you can be able to assist knowledge transforming to intellectual property rights, and then the licensing techniques should develop, meaning all the business management, the intellectual licensing techniques and branding and business financing and so on, so that you can be able to have entrepreneurs accessing these on the basis of some kind of agreement. So there are things that are going on, but certainly there's a lot of synergy, cooperation, and partnership required, not just between the institutions, but between the very, very powerful knowledge workers, the researchers, and the individuals, to make sure that Nigeria realizes that unless our knowledge is transformed to our solutions, we will continue to have this very high poverty rate in our country. Thank you.
Please, Professor Gunda, did you have anything in addition to answering the question? You can spend 1 minute to add your voice. Thank you. Unmute yourself, Prof.
Yeah, I wish the organizers, it has started very well with the keynote address, and some of the questions that have been thrown up. And we've heard about the state-of-the-art researches to solve pharmaceutical problems. I think towards your communique, people should deliberate on some of these points. And if you can come up with an answer that will help us, it will be very good. Because I believe that we now need to have a restructured approach to our research, but it has to be led, both financially and leadership. There's a lot that has been done, but how do we translate it? I pray that whatever we come up with will be useful for the country. Thank you.
For that addition, very insightful. So in preparation for the parallel session, we are going to take 2 oral presentations. This conference is in 5 tracks. We have clinical pharmacy, pharmaceutical chemistry, pharmacognosy, pharmaceutics and pharmaceutical technology, pharmaceutical microbiology, and biotechnology. Please, I want you to add a suffix to your name in preparation of the breakout section while the oral presentation is going on. So now we want to know if poster number 43 is still on this call. Ewuru et al. And Elaribe et al. Poster number 78. So we're going to play the pre-recorded oral presentations. We're going to play them, except we are starting with abstract 78, Alaribe et al. We are playing that oral presentation. Thereafter, we'll play poster abstract number 43. And the questions will come together because we are mindful of time. Thank you. The floor is yours, Dr. Larry.
So welcome. I am Blessing Elijah from the Department of Pharmaceutical Chemistry, Faculty of Pharmacy of the University of Lagos. Today, I will be making a presentation on the synthesis, bioimaging, and evaluation of the safety of the extract and nanoparticles of Abrus precatorius. The aim of this research is to synthesize nanoparticles using the seed and seed coat of Abrus precatorius and evaluate the safety dose of the nanosynthesized molecules by the assessment of toxicity in target organs after a repeated administration in a mice model. As an introduction, numerous natural products have been established to possess therapeutic effects, but have often been found to exhibit obvious toxicity at their effective doses. This major reason has prevented several potential leads from proceeding to clinical trials. For instance, studies have shown that various parts of Abrus precatorius possess pharmacological activities such as antiviral, antioxidative, anticonvulsant, anti-cancer, anti-malaria, and so on. Nanoparticles, due to their high surface area to volume ratio, are able to exhibit unique features, for instance, cellular penetration. Therefore, they are able to achieve the effects of macro-scale bulk materials at lower doses. Also, recently, digital image analysis has been incorporated in the diagnosis of pathological conditions because it affords a better assessment of cellular, tissue, and organ toxicity. Here is an overview of the methodology used in this study. The seed of Abrus precatorius, also known as rosary pea, was utilized in this study, as indicated by the green arrow at the top left quadrant of the slide. To begin with, the seed was separated from the seed shell and both samples were pulverized, followed by aqueous extraction. Each filtrate was freeze-dried and was subsequently used for the nanoparticle synthesis. This involved the reduction of silver nitrate by compounds present in plant extracts. The nanoparticles were labeled B1 for the seed nanoparticle and A1 for the seed coat nanoparticle. The crude extracts were labeled B2 for the seed and A2 for the seed coat, as shown in top right quadrant of the slide. Intraperitoneal median later dose validation was carried out, followed by dose selection, animal grouping, and a 28-day interperitoneal administration of the samples. Toxicity of the 4 samples were compared with negative control group on the basis of clinical signs of toxicity, histopathological examination, hematological analysis, and digital bioimage analysis. From the results, the animals which received the mid dose and highest dose of sample B2 died within the first week of the study. Prior to their death, they showed clinical signs of toxicity such as weight loss, marked loss of appetite, inactivity, rough air coat, and diarrhea. The body weights and percentage body weight changes of all the surviving treated animals were not significantly different when compared with the negative control group. However, the result of hematological analysis showed that the number of platelets was lower for the 4 samples as compared with the negative control. It was also observed that as the doses of samples A1 and A2 increased, there was a corresponding decrease in the hemoglobin, red blood cell count, and hematocrit of the treatment groups. Meanwhile, sample A1 treatment group showed greater response in hematocrit with increasing dose, as shown in figure 5. Likewise, white blood cell parameters such as white blood cell count, lymphocytes, and granulocytes increased with increasing dose of sample A2. There was also a reduction in the average volume of the red blood cell indicated by the mean cell volume with increasing doses of sample B1, which was in contrast with sample A1, which showed a decrease. From the histopathological evaluation, the liver was the most affected of the 3 target organs examined in all the treatment samples, followed by the heart. The kidney was the least affected. For instance, both A1 and B1 treatment groups, that's the seed and the seed coat nanoparticles, had normal kidneys at the lower and mid doses, but only showed slight renal toxicity at the highest dose. The crude extract of B2, that's the seed sample, showed toxicity even at 0.07 milligram per kilogram. In terms of Bowman's capsule morphometry, sample A1 had the most significant difference from the negative control at all doses. Meanwhile, at a dose of 0.60 milligram per kilogram, sample A2 could reduce the size of glomerulus significantly and thereby increase the Bowman space, and this could indicate glomerular atrophy. None of the measured Bowman's capsule's morphological parameters of sample B1 showed a significant difference from the negative control. Results of the Bowman's capsule morphometry in sample B1 are shown in the next slide. Though it was observed that the mid dose of sample B1 had a lower glomerular area of Bowman's capsule, and Bowman space, as well as Bowman's capsule perimeter, the difference was not of significance upon statistical analysis. In conclusion, these studies showed that synthesized nanoparticles from seed and seed coat of Abrus precatorius had significantly lower toxicity than the crude extracts. Meanwhile, at doses lower than 0.07 milligram per kilogram, the synthesized nanoparticles could be used as safer alternatives to assess the various pharmacological activities of the plant parts. I would not end this presentation without acknowledging my research partners, headed by Dr. Chinwe Stephanie Alaribe. Our words of encouragement and genuine push for overall excellence were significant catalysts for the success of the research. I also appreciate Mr. Daniel Usiago of the Anatomy and Molecular Pathology Department for his expert advice and introduction to practical light microscopy. I appreciate Dr. Sunday Oko of Federal Institute of Industrial Research for his immense contribution. I would like to acknowledge the support of my undergraduate research partner, Olorun Toba Ayodeji. As they say, 2 heads are better than 1. Thank you.
And now we call on abstract number 43. Please get your questions ready for abstract number 78. We'll take the questions together. Thank you. Abstract number 43, the floor is yours.
Good morning, distinguished participants. All protocols are duly observed. My name is Nkechi Enwu of the Department of Pharmaceutical Microbiology and Biotechnology, Faculty of Pharmacy, University of Lagos. The title of my presentation is Microencapsulation of Probiotic, Evaluation for Viability and Cytotoxic Effect of its Postbiotic Metabolites on MC7 breast cancer cell line. Background of the study. Probiotics are components of bacteria that have shown to produce beneficial effects on human health. Substantial evidence had it that probiotic alleviates various ill health conditions, including suppressing cancer cell growth. Cancer remains one of the most common causes of death worldwide. The standard cancer medication often fail to achieve complete cancer remission due to site of tumor growth, stage of the cancer, metastasis, and non-specific toxicity to normal cell. Thus, an urgent attention is needed. Microbial-based therapy prevents and destroys cancer cells. It is derived from naturally occurring live, genetically modified, or purified product of microorganisms that specifically target cancer cells, limits systemic pathogenicity, and then boosts anti-cancer efficacy. However, cell viability is often low due to harsh conditions exposed during processing, handling, storage, and gastrointestinal transit. Therefore, microencapsulation is a promising technique that protects against adverse conditions to which probiotics can be exposed. Aim of the study. The study is aimed to formulate encapsulated probiotics using spread drying method. Objectives of the study: to isolate and identify probiotic microorganisms, harvest its postbiotic metabolites, investigate its cytotoxic effect against MCF7 breast cancer cell line, encapsulate the probiotic bacteria, evaluate their viability in simulated gastrointestinal fluid, and during storage for 12 weeks at different temperature. Methods. A total of 25 human and animal milk were collected and immediately delivered in the lab and stored at 4 degrees Celsius. Samples were analyzed by serially diluting and then inoculated in sterile MRS agar, incubated anaerobically at 37 degrees Celsius for 72 hours. Isolated bacteria were identified by conventional and then Vitek2 method as previously described. Cytotoxic tests. Postbiotic metabolites were extracted from centrifuge supernatant of 24-hour bacterial culture. The collected sample were placed in 2 sterile test tubes labeled K1 and K3. The pH of K1 was adjusted to 7.4, while the pH of K3 labeled was 4.8 and was left unadjusted. Negative sample labeled K2 was sterile MRS broth, while positive control sample was 5-fluorouracil and cisplatin. The breast cancer cell line was purchased from Germany, grown on Delbecker modified media methods using hemocytometer. The harvested sample was seeded in 9 cyst well plates and then incubated for cytotoxicity assay as previously described. Encapsulated of probiotic. The bacterial cell was concentrated using centrifugation, and plate count was used to produce 1 × 10⁻⁹ colony forming unit per mil for microencapsulation. Probiotic was formulated by using method previously described. Table 1 is the composition of formulated microcapsule. The property of the microcapsule was evaluated, such as the percentage yield, bulk density, disintegration time, and weight uniformity. Viability test. Survivability of the free and encapsulated probiotic was evaluated in simulated gastrointestinal fluid and during storage for 12 weeks at different temperatures as previously described. Results. Lactobacillus plantarum was identified from animal milk. Fig 1 is the breast cancer cell line under microscope. The cell was 99.7% viable before cytotoxic test. Fig 2 is the bar chart results of cytotoxic effects of the test sample on breast cancer cell line using the concentration of 0 to 25%. Y-axis is the baseline for measurement of cytotoxicity. Values below 100 shows cytotoxic effect, while values above 100 represent lack of cytotoxic effects. X-axis is the postbiotic concentration from 0 to 25%. Table 1 is the inhibition concentration of 50% growth as calculated by GraphPad's Microsoft version. Fig 3 is the picture of microcapsule granulus and 500 milligram capsule shell. And table 3 is the result of properties of microcapsule. Table 3 is the survivability of free and encapsulated cells in simulated gastrointestinal fluid. Our results show that free cells rapidly reduced in viability in simulated gastrointestinal fluid. Fig 4 and Fig 5 is the survivability of the free and encapsulated cells during storage at 4 degrees Celsius and 25 degrees Celsius. Our results show that encapsulated cells achieve highest survivability during storage for 12 weeks at 4 degrees and 25 degrees Celsius compared to free cells stored at 4 degrees and 25 degrees Celsius. Conclusion. Lactobacillus plantarum probiotic exhibits cytotoxic effects on breast cancer cell. The postbiotic could be used as combined therapy at temperature. Below is my references. Thank you.
Thank you so much, Ehuru Ethel, for that presentation. Please, if you have questions, you are free to raise your hand. I'll put your question in the chat box for both abstract number 078 and abstract number 04. Thank you. Put your questions in the chat box. In the absence of you are still waiting, please, for your question. Kindly ask and specify which abstract you are referring to. The first speaker was abstract number 78, and the second speaker was abstract number 46. Please, your question. Thank you. And like we said, if you cannot join the breakout session, put the prefix to know the track you are attending for the breakout session, for the parallel session. So for pharmacognosy, put PCG before your name. For clinical pharmacy, you put PCL for your name. For pharmaceutical microbiology and biotechnology, you put PMB. For pharmaceutical chemistry, you put PCH. And for pharmaceutics and pharmaceutical technology, you put TC. This is just for us to put you in your specific room if you cannot join the breakout session yourself. Questions for the oral presenter. In the absence of any question, we are now moving to the parallel section. Kindly join one of them. Join the breakout rooms for the poster session. Thank you.